Ashani Weeraratna, who is an outstanding scientist and a dear friend. And I'll give a little background about Ashi's current many titles and where she developed from as a scientist. So Ashi is the E.V. McCollum Chair of Biochemistry and Molecular Biology at Johns Hopkins School of Public Health. She's also a Bloomberg Distinguished Professor, co-leader of the program in cancer invasion and metastasis at the Sidney Kimmel Cancer Center. And I had interacted also a lot with Ashi before joining Hopkins, where she was a named professor and a program leader at the Wistar Institute in Philadelphia. Ashi has a very international background, having been born in Sri Lanka and grown up in South Africa. I think a brother who lives in Scotland. Ashi is just a very cool person with a great background and great science. And one of the things that I would really emphasize about Ashi is that there's been a real emphasis and an appropriate emphasis on supporting junior faculty, female faculty, and scientists of color over the last few years. But I can say that I can think of pretty much no one who is more of an advocate for all of those areas over a decade ago already, and has been tireless in her efforts in promoting that. And it's really great to see that the rest of the world is slowly catching up on those fronts. It's a real great pleasure to hear, and I know a fair amount about the science that she's going to talk about, but it's a really appropriate area, especially at Yale, where we're thinking about having a new aging center based in the pathology department. And I think Ash is going to be talking about age against the machine, how the aging microenvironment governs tumor progression and response to therapy. I was thinking of playing some Rage Against the Machine in the background during the introduction, but I thought that'd probably go horribly wrong. So you'll just have to imagine that. And Ashi, it's a real pleasure having you be a Yale SPORE and Skin Cancer Center Ground Round speaker.
Thank you so much, Marcus, for that lovely and warm introduction. And yeah, the title appeals to a very specific demographic of people, I think. So thank you so much. I'm delighted to be giving this talk, although I really do wish it was in person. And I will be talking to you all, as Marcus said, about our work in the aging microenvironment and how that governs response to therapy. So we've been so interested in aging as a driver of tumor progression, because we know that it's one of the most significant prognostic factors for the development of cancer. So of all 100 people diagnosed with cancer, 89 of them will be age 50 or over. And of those that are diagnosed with cancer and die from this disease, again, the largest percentage of them is over the age of 50. And so we've been trying to understand why that is and why that happens. Obviously, there are a lot of systemic factors that drive tumor progression, but we've been super interested in what is happening in the local microenvironment, specifically of melanoma. But as I'll tell you later, we are expanding into other cancers such as pancreatic cancer as well. So what we have seen in our studies is that there are significant changes that occur, largely due to fibroblasts, and I'll tell you a bit more about that in a second. And these changes can affect not only the way tumor cells grow, but the way endothelial cells grow into the tumor, as well as the biophysical matrices around these tumors, allowing them to metastasize and invade more effectively with age. So a few years ago, what we did, and this was a very collaborative piece of work, and Marcus was on this paper as well. The cell lines and resources that he's developed have been absolutely critical for us and continue to be to this day. So we're very grateful for those. And the reason that we looked at the skin was that we were very interested in the fibroblasts themselves, because fibroblasts in the skin, also in work out of Yale from Valentino Greco's lab, tend to age with the individual rather than undergo a lot of turnover. And we were very curious to know, as they age, what were the differences they were secreting and how were they changing their physical environment, because the melanoma cell spends much of its life right here at the intersection of the epidermis and dermis, becoming invasive. So in order to recapitulate that work, what we did was to take skin fibroblasts from the upper inner arm, so intermittent sun exposure, of individuals, healthy non-melanoma bearing individuals in their mid-20s to mid-30s, and then in their in the age where sort of melanoma starts, the incidence of melanoma starts to skyrocket, which is 55 to 65. We use those fibroblasts to create artificial skin, which is a technique taught to us by our friend Meinhard Herlin. And in doing that, we were able to see that when we made reconstructs with fibroblasts from young individuals versus fibroblasts from aged individuals, those melanoma cells would invade far more rapidly in the fibroblasts, in the skin reconstructs made with fibroblasts from aged individuals. Now, the only difference between these sets of skin reconstructs is the age of the fibroblast. Everything else is the same. So we wanted to know if we could recapitulate that in vivo. And as I mentioned, Marcus, along with Martin McMahon, has made the BRAF P10 mouse model of melanoma. Marcus then backcrossed these to C57 black 6 mice. And as I'm sure you all know, created a series of cell lines. Most of the experiments I'm going to show you are from the Yum 1.7 cell line, which we have taken and then injected into either young mice of 6 to 8 weeks of age, or age mice of 12 to 18 months of age, depending on the experiments we're doing. And what we saw was that actually in the young mice, the tumors grow much faster and they grow much more slowly in the aged mice. However, they metastasize to the lung far more effectively in the aged mice than they do in the young mice. And so we wanted to better understand that. And Mitchell Fain, who is a postdoctoral fellow in my lab, a very talented postdoctoral fellow in my lab, decided to do an experiment where he seeded the Yum 1.7 cells intradermally in the skin of the mice, and then he allowed them to metastasize over time to the lungs of the mice. And at 3 weeks, he took the lungs of both the young and the aged mice, and he looked for his M-cherry labeled melanoma cells. And what he found was that in both the young and aged mice, there were these sort of single or maybe double cell colonies all over the lung. If he waited just a couple more weeks, and he did this at 5 weeks, what he saw is that in the young lung, the colonies remained as these single cells or much smaller colonies, whereas in the aged lung, they had started to grow out quite dramatically, and we could quantitate this over a series of mice. Mitch wanted to know what kind of role the fibroblasts played in all of this. And so he took fibroblasts from the young lung and from the aged lung, and then he seeded GFP tagged melanoma cells in a 3D sandwich with these fibroblasts. And what he saw is that when he incubated melanoma cells with aged lung fibroblasts, they would proliferate far more rapidly than when he seeded them with young lung fibroblasts. He then compared the growth rates of the melanoma cells with lung fibroblasts to that of the skin, and he found that the aged skin fibroblasts actually suppress the growth of these melanoma cells, which we had seen before and which I just showed you in the mouse model, whereas a young skin fibroblast promoted it. And so we saw a distinct difference between the way the fibroblasts in these two different tissues behaved, which was sort of a very striking and eye-opening thing for us. So Mitch then did proteomics on both the skin fibroblasts and the lung fibroblasts, comparing the age to the young in both cases. And what he found was something quite interesting. He found that in aging skin, there was a signature of fibroblasts promoting a non-canonical Wnt signaling phenotype, which included genes like SFRP2, SERPINE2, DKK1, Wnt5A, and RR2. In the aged lung, however, he saw a signature that showed there was a promotion of canonical Wnt signaling. And what he would see is sometimes the same family members, SFRP1 and SFRP2, which we know to play very different roles in Wnt signaling, were the ones that were differentially expressed. And so I'll tell you a little bit more about these two guys. So we had shown a few years ago that aged fibroblasts secrete SFRP2. And when they do that, they shut off beta-catenin signaling. So SFRP2 inhibits canonical Wnt signaling. And in doing so, decreases the ability of a melanoma cell to react to the reactive oxygen species in the microenvironment because it disables its base excision endonuclease repair gene AP1. Sorry for the inappropriate animation over here. We'll get to it in a second. However, so what that did was to decrease the proliferation of the melanoma cells, but make them more invasive. However, when Mitch looks at SFRP1 and he treats melanoma cells with recombinant SFRP1, they increase their proliferation and they actually shut off non-canonical Wnt signaling. In what Mitch then did was to do an experiment in vivo where he took age mice. He allowed 3 weeks for the initial dissemination of the tumor, as I showed you previously. And then once the tumor cells had seeded in the lungs, he treated the mice with antibodies against SFRP1. So in the IgG control, you see this outgrowth of metastases, as I showed you earlier. But in the mice that were treated with anti-SFRP1, you see that the cells that have seeded in the lung remain there as single cells, which was super interesting to us. So the reason this was so interesting is we've been working for a while on this idea of what we call phenotype switching, where we have canonical Wnt signaling that's driven by beta-catenin and non-canonical Wnt signaling driven by Wnt such as Wnt5a. And we had always associated the Wnt5a phenotype with metastasis and the beta-catenin phenotype with proliferation. But what I'm going to tell you shows that we were not as sophisticated in our thinking as we should have been. And instead, the roles are much more interchangeable and much more complex. So important to note that Wnt5a promotes an invasive but slow cycling phenotype. And that led us to wonder whether these changes we were seeing in the young versus aged lung colonies had any, I'm sorry, I don't know what's happening to my animation, had any relation to dormancy. And so we turned to our good friend, Julio Guerra-Guiso, who's now just started an Institute of Dormancy at the Albert Einstein College of Medicine in New York. And he is a world leader in understanding tumor dormancy, and he has these signatures of dormant genes. So what Mitch did was to look at the expression of these genes in Wnt5a high versus Wnt5a low cells. And what he sees is that Wnt5a high cells carry very strong markers of dormancy, whereas Wnt5a low cells carry very high markers of proliferative cells. And so Mitch's question was, does the aging microenvironment drive a switch from a Wnt5a high to a Wnt5a low phenotype, and in doing so drive increased proliferation in melanoma cells? So to answer this, the first thing Mitch did was to take melanoma cells and expose them to the conditioned media of young and aged lung fibroblasts. And what he sees is that indeed in the same melanoma cells exposed to age conditioned media, these are just three separate, these are the same melanoma cells, three separate donor fibroblast media. What Mitch saw was that the markers of dormancy were decreased when he exposed melanoma cells to these age-conditioned media from the lung, whereas the markers of proliferation were increased in the melanoma cells. The next thing he did was to look at Wnt5a specifically and to look at it in vivo. And what he saw is that if he stained young and aged tumors for Wnt5a, and Ki67 in the lung. So these are cells that he has implanted in the skin of the mice that have now metastasized to the lung. They're labeled with M-cherry in the absent, and this is just staining for M-cherry, showing you that there are far fewer cells in the young lung than there are in the aged lung. And if he stains the lungs for Wnt5a, these large tumors that are growing out in the aged lung have much less Wnt5a staining than the tumors in the young lung, and they're highly positive for Ki67, telling us that the Wnt5a may be driving this dormant phenotype in the lung, which was super interesting to us. What Mitch did then was to manipulate Wnt5a in these conditions. So he took young mice and he injected cells with a DOX-inducible SH-Wnt5a. So he knocked Wnt5a out of the melanoma cells in the young mouse lungs. And what he saw is if he did that very early on, he could reduce the number of metastases altogether. But if he did that later, he could cause the metastases to grow out. If he did the opposite experiment where he took aged mouse lungs and then he gave them Wnt5a, he could actually prevent these tumors from growing out at day 21. And he could also, if he induced the Wnt5a at day 3, they had already started to get to the lungs, but they again were prevented from growing out. So this was absolutely fascinating to us because it kind of changed our thinking of how Wnt5a was driving metastasis. And what we saw was that, these melanoma cells in the young, first of all, the tumors are much bigger in the young skin than the aged skin. And so even though we might see similar rates of seeding in the lung, we know that the rates of tumor cells leaving the aged skin are higher than the rates leaving the young skin. But once they get to the young lung fibroblasts are secreting factors that maintain the Wnt5a phenotype and retain those cells in this invasive but slow cycling state. However, in the aged lung, we're seeing that there's an increase of secretion of SFRP1, and that is allowing these cells to now lose that slow cycling state, become more proliferative. These are also positive for beta-catenin, MITF, and MART1, and they're rapidly proliferating. And so really for us, where we had always thought of Wnt5a as simply a driver of metastasis, it's actually playing a much more complicated role and driving an invasive but then dormant tumor phenotype that requires a change for these cells to come out of dormancy. I will add that we've also seen changes in the immune microenvironment in both the young and aged lung that are contributing to this outgrowth and lack of immune editing of these cells as they grow out. So, sorry, hold on a second. So I've started to give you now a snapshot of the fact that aging can drive metastasis of tumors, but we've been very interested in also all of the other things that the aging microenvironment can do, from driving not only metastasis, and we'll talk a little bit more about this, but things like therapy resistance, angiogenesis, metabolism, and changes in the immune microenvironment as well. So I'll start with the angiogenesis story, which is a story that was recently published out of our lab. I should mention that all the work I just showed you is of Mitch's is completely unpublished at this time, and most of the slides I'll show you today are unpublished work. But I thought I'd give you some snapshots of some recently published work as well. So the tumors that we grow in aged mice have far more angiogenesis if we stain with either CD31 or even CD105. And when we take dermal microvascular endothelial cells, and we treat them with medium from young and aged fibroblasts, we see that those dermal microvascular endothelial cells will form networks when they're treated with age-conditioned media, but not so much when they're treated with young. And we can quantitate this as well. And this was really mysterious to us because we knew that VEGF and its receptors were decreased during aging. And so it didn't make sense to us that we were seeing a decrease in VEGF, but an increase in angiogenesis. However, we knew from our work with SFRP2 that SFRP2 has been shown to stimulate angiogenesis via a Wnt-related signaling pathway. So Wnt keeps rearing its head again. And we knew that if we treated mice with recombinant SFRP2, we could increase the metastases of these cells. So Mitch, along with Brett Ecker and Aman Kar, decided to explore this further. And what they did was to take these endothelial cells, treat them with either recombinant SFRP2 in young media, or I don't know why that keeps happening, or an antibody against SFRP2 in age media. When they manipulated SFRP2, they could show that when they increase it, these microvascular endothelial networks increase. If they decrease SFRP2, they can disrupt the formation of networks. They also did this in vivo and showed exactly the same thing. If they give young mice recombinant SFRP2, they have a ton more angiogenesis. Old mice have more angiogenesis, but if you treat with an antibody against SFRP2, it decreases the number of blood vessels. And so when Mitch and his colleagues looked at VEGF and SFRP2, what they found was that these young mice had very high levels of VEGF, but the tumors in age mice did not. The opposite was true for SFRP2. And so, that led us to ask the question, what does that mean for anti-angiogenic therapy? Because anti-angiogenic therapy, of course, is designed against VEGF. And so this hinted to us that younger patients might benefit from this therapy, but certainly older patients who had highly angiogenic tumors that were not dependent on VEGF may not benefit. So to answer that question, what we did was to turn to our colleagues Pippa Corey and Mark Middleton in the UK, who had just conducted this large trial for Avastin and melanoma, where they had treated over 1,300 patients. Their observation at the end of this trial was that overall, there was no change or no response to Avastin. However, we asked them to go back and reanalyze their data, and this time stratified by age. And when they do that, sorry for the traffic outside my window. And when they do that, we see that patients under the age of 45 who receive Avastin actually do better on Avastin, whereas those over the age of 65 and older really have no difference in their response to Avastin. To sort of close this loop, what Mitch then did was to take young animals, he treated them with an antibody against VEGF, and then attempted to do that in the presence of high levels of SFRP2. And so when he does that, there's no change and no response to the mouse equivalent of Avastin in tumors in which have high SFRP2, which may be the reason why we're not seeing older patients responding to this therapy as well. So one of the things that we learned from the study was that, not only could SFRP2 take over from VEGF as a driver of angiogenesis during aging, meaning that older patients, were unlikely to respond to Avastin, the reviewers had actually asked us some questions about the matrix and what was happening to the permeability of these blood vessels. And so although we felt that it was out of the scope of that particular paper, it was a question that really intrigued us. And so my graduate student, Gloria Marino, decided to take this on. The reason we found this so interesting is because of a previous study from Aman Kar in my lab, who had shown that collagen density is decreased during aging. And that can happen whether it's in the presence of a tumor or even altogether in the absence of a tumor. So this is just normal mouse skin from an 8-week-old compared to a 12-week-old mouse. And I think you can see that the collagen looks dramatically different between the two. And Aman wanted to know what was driving these differences in collagen. She identified this protein called haplin-1, which was actually the protein that was the most significantly increased in the young skin fibroblast secretome. And haplin-1 turns out to be a super interesting protein because it's the protein that knits together the collagen and the elastin in the skin and maintain sort of the integrity of the skin. So, when you're young, you have this lovely smooth skin. And as you age, those collagen and elastin crossings break down, and that's a little bit how wrinkles occur. And so haplin-1 is responsible for stitching together hyaluronic acid to proteoglycan monomers. So Aman decided to explore this, and the first thing she did was to just simply inject it into mouse skin and see what it did. And she found that if she put it in aged mouse skin, she could redensify the collagen again in the aged mouse skin. She wanted to know what that meant for the type of fibers that these fibroblasts were laying down in the ECM networks, the extracellular matrix. And so we collaborated with my dear friend, Itika Kierman at Fox Chase, and Aman seeded fibroblasts. And then what she did was to basically look at the matrix they left behind and look at the orientation of those fibers. And when she does that, she sees that with young fibroblasts, the fibers are oriented in multiple different directions. And each direction is assigned a color. So you see this very colorful matrix. If she knocks down haplin-1 in these fibroblasts, she now sees that the direction of the fibers is more aligned, fewer colors, means fewer directions of the fibers. And if she reconstitutes this by adding back haplin-1, she can start to increase the multi-directionality of these fibers again. We can do the opposite experiment in the age. So you can see that the age fibroblasts start out looking very linear. And if we add in recombinant haplin-1, we can now increase the multidirectionality of the fibers. If we first boil the haplin-1 before adding it in, it doesn't do that. So it tells us that it really requires a haplin-1 activity. Aman wanted to know what that meant for the invasion of the melanoma cells in vitro. And so she added in recombinant haplin-1 into reconstructs made with aged fibroblasts, and showed that when she does that, they're no longer as able to invade as effectively into the membrane. If she does the opposite, where she depletes haplin-1 in the fibroblasts before making reconstructs with them, they increase their ability to invade into the membrane. If we do this experiment in vivo, where we treat the primary tumor with haplin-1 in the age mice, we no longer see these cells able to metastasize to the lung in the age mouse versus those mice treated with haplin-1. And so we were super excited by those data, and even more so when in a parallel study, Brett Ecker, who was a visiting clinician to the lab, showed that haplin-1 played a role not only in the metastasis of these cells from the primary tumor, but also the haplin-1 played a critical role in maintaining the integrity of the extracellular matrix around the lymphatic vasculature and the lymph node as well. And when haplin-1 broke down during aging, to primary tumor cells leaving the primary tumor site could escape both through the lymphatic vasculature and not spend too much time in the lymph node, but go on to very quickly form visceral metastases. So that was one of the studies that first showed us that this loss of integrity of the ECM during aging might actually help to direct the route of metastatic dissemination from the primary tumor. So taking all of those data, the angiogenesis data and the matrix data together, Gloria Marino, who's a grad student in the lab currently, decided to explore this further. And what she saw was that if she stained for blood vessels, they were sitting in very different matrices in aged versus young skin. And again, to remind you, this is what the aged versus young skin looks like. So she wanted to know whether that difference in these matrices could impact angiogenesis. And so she embedded her endothelial cells in a matrix that had aged or young fibroblasts, or aged fibroblasts treated with recombinant haplin-1, or young fibroblasts in which haplin-1 had been knocked down. What Gloria saw was that the endothelial cells in the H matrices had all of these sprouting, these are basically what we think of as little artificial blood vessels that are sprouting off the endothelial cells as compared to the young. And if she treats the aged endothelial cells with the endothelial cells in the H matrix with haplin-1, they decrease their ability to sprout. And if she knocks down haplin-1 in the young fibroblasts and then embeds endothelial cells there, they increase her ability to sprout. So haplin-1 was having a direct impact on angiogenesis. And so when she looked at mouse tumors, she saw the same thing. Again, there's more angiogenesis in the age versus the young. But if she treats the age tumors with haplin-1, she can reduce quite dramatically the amount of angiogenesis ongoing. What was interesting, though, is that when we stained for V-cadherin, we saw something quite different. Again, there are far more blood vessels in the tumors in the aged mice. However, they don't stain very well for V-cadherin. They stain beautifully for CD31, CD105, etc. But the V-cadherin staining is super weak compared to the young or the aged plus haplin-1. So Gloria wanted to understand that better. And our hypothesis was that young fibroblasts lay down a matrix that endothelial cells can anchor to really beautifully, and that sustains the interactions between their cells as well. However, aged fibroblasts, that matrix is disrupted, disrupting the integrin connections between the age matrix and the endothelial cells, and we hypothesized that the cell-cell interaction, specifically V-cadherin, would also be disrupted. And so that's exactly what Gloria sees. So she laid down endothelial cells on matrices that she had made from young or aged fibroblasts. You can see that the endothelial cells on the young matrices have beautiful V-cadherin, but on the age, they lose these connections. And now she wanted to manipulate Haplin-1 to sort of knot up these matrices and see what happened. If she knocks Haplin-1 out of the young fibroblasts, they now lose their ability to make these nice V-coherent connections. And if she adds Haplin-1 into the H fibroblast, now the endothelial cells have beautiful V-coherent connections. So that was super exciting, but she also wanted to know, did that mean that if they had these nice tight V-coherent connections, was there barrier integrity of these endothelial cells? And to measure that, what Gloria did was to use an electrode assay where she seeded the CDM, so the fibroblast-derived extracellular matrix, and then she plated the endothelial cells on top of that matrix. And then she measured the current. So the more resistance there is, the better the barriers are and the tighter these interactions are. So in endothelial cells seated on an HECM, there's very little barrier integrity. On those seated on a young CDM, there's a lot of barrier integrity. And if we take our cells that are on an HCDM and we give them Haplin-1 to knot up their matrix, we can now see that there's an increase in barrier integrity. So basically, all of these data from Gloria so far are telling us that the more Haplin-1 there is, the tighter these matrices are, the more intact these blood vessels are as well, which can have significant impact for tumor cells being able to invade in and out of that. And so now what Gloria is doing is some very beautiful in vivo imaging of these vessels and of the flux of tumor cells in and out of these vessels in these different conditions. So I'll move on to the next story that we recently published. So this was work conducted by Gretchen Eliseo, who was a grad student in my lab at the time. And what she did was to notice that H fibroblasts made a ton of lipids, and they secreted a lot of these lipids. And when they secreted a lot of those lipids, melanoma cells would take those lipids up. And so these are melanoma cells grown in young or age-conditioned media, and then simply stained for Bodipi. And if Gretchen looks at the lipidomics of what is being secreted by the young or H fibroblasts, she can see that whatever H fibroblasts are secreting, melanoma cells are taking up. So that was super interesting. And there were two questions. One, how are they taking it up? And two, what are they doing with it? So Gretchen looked at a bunch of different fatty acid transporters and identified this particular one called FATP2. And FATP2 is increased in melanoma cells exposed to age-conditioned media. It's increased in melanoma cells grown in skin reconstructs with H fibroblasts. It's increased in melanoma cells that we put in H tumors, in H mice. If we look at patient tumors, this is just TCGA data, we can see that patients over the age of 50 are the ones who have the most FATP2 expression. So the other thing that we noticed is when we were staining patient tumors for FATP2, we noticed that the patients who survived the shortest time after being treated with BRAF/MEK inhibitors were patients who had very high FATP2. So that made Gretchen ask the question, could FATP2 and lipid metabolism be playing a role in therapy resistance? So something we've seen before is that patients over the age of 65 have a lower response. These data in this slide are single agent. I'll show you double agent in just a second. So this is just vemurafenib, and these are data from the very early trials. And we found that patients over the age of 65 in those trials were less likely to mount a complete response to BRAF inhibitors than patients under the age of 65. And in our mouse studies, we showed that the exact same tumors implanted into young mice would respond to the vemurafenib tool compound, whereas those planted into age mice would not. So Gretchen wanted to know how FATP2 could affect this, and she wanted to do this using the BRAF and MEK inhibitors. So she created a cell line in which she had knocked down FATP2 in a DOX-inducible manner. And what she found is that when she knocks down, so she has an empty vector control, and then she has the empty vector plus the BRAF/MEK inhibitor. And in tumors in young mice, of course, they respond to the BRAF/MEK inhibitor, and after some time they grow back. So we've all seen this a million times. If she now knocks down FATP2 using dox and treats with the BRAF MEK inhibitor, it's exactly the same in the young mice. The tumors respond, they eventually grow back. However, in the age mice, it's a completely different story. What we see is that the tumors, first of all, treated with BRAF MEK inhibitor in the age mice kind of just stop growing for a little bit, but then continue to grow, so they rarely respond at all. But now as she first treats them by knocking down the FATP2, you can see that those tumors basically go into remission and stay remissed, if you will, for a very long time. So this was super exciting data to us because it was one of the first incidences we really had of targeting this very age-specific change in a melanoma cell and showing that we could overcome therapy resistance quite dramatically in this case. So, you know, a lot of times I think that the questions are my favorite part of a talk because they make me think and they make me think about what we want to do in the future. And out of the questions have come some questions about, well, you see a lot of changes with age. Does gender, or I guess to be technically correct, biological sex play a role? And so this is something we've just started exploring in the lab. We see that in melanoma, there's a big difference in mortality estimates in males versus females, as well as differences in incidence. And so Yash Chabra, who is a junior faculty in my lab, started to explore this. And what he found was that while there are certainly differences between male and female, they tend to be less qualitative and more quantitative. So for example, if we look at something like senescence, senescence increases with age in both female and male dermal fibroblasts, but it increases to a higher extent from the start point in the male fibroblasts versus the female fibroblasts. If we look at changes in lipid oxidation, we see the same thing. And if we look at changes in things like exosomal content, so this is work done by Laura Hooser, who is also a postdoc in the lab, we see that, again, between males and females, there are distinct differences in the changes that we see in CD9. So CD9 is decreased in both males and females during aging as compared to the young exosomes. This is an ExoView chip. But we see that in the males, it's far more dramatically decreased than it is in the females. If we look at the impact of these fibroblasts on melanoma cells, we see the same thing. So we see that if we treat melanoma cells with the BRAF MEK inhibitor, this is a spheroid assay where we're just looking at survival. We see that melanoma cells treated with BRAF MEK inhibitor in the presence of aged male conditioned media do not die as effectively as they do when they're treated with young male conditioned media. So the more red you see, the more dead cells there are. And the same is true for melanoma cells treated with aged female conditioned media, but again, the impact is not as great. So you can see that quantified here. Melanoma cells treated with aged male conditioned media have far less relative cell death than those treated with aged female conditioned media. The same is also true for invasion. So in vitro, at least, we see that there is an increase in invasion in melanoma cells treated with aged males conditioned media versus aged female conditioned media. What's been fascinating is that recently we've been able to get fibroblasts from the same people, so they're genetically identical, and they've been collected 20 plus years apart. And what we're seeing is that even within the same individual, that's now reflecting some of the changes we see. So that's been super exciting. So here within the males, you can see that there is a distinct impact in the increase in invasion. This is two different men. The fibroblasts taken over 20 years apart in each case. In the females, again, the trend is there, but it's not as dramatic as it is in the male. And that's sort of a constant theme that we see here. Yash also did the in vivo experiment where he took both the YUM cells, which are male, and these BSC9AJ2 cells, which are female. And he sort of did the crisscross experiment where he put them both in male and female, or both in, and these both in male and female. And what he sees again, over and over again, is that When he puts the YUM cells in young versus age male, again, they grow far more slowly. But if he does this in females, in the YUMs, there's a little bit of a difference, but in the female to female, there's very little difference. Although again, there is a big difference between the way the age males grow. If he looks at Ki-67, he sees that the tumors are proliferating far less in the age male mice than they are in the age female mice. So telling us again that there are distinct differences in the microenvironments between the male and female, and we're still trying to figure out exactly what those differences are. We see that there are changes in angiogenesis. Again, the same story. There's more angiogenesis with aging, but again, it's more dramatic in the males than it is in the females. If we look at metastasis, we see the same thing where we have more metastasis in the males. Actually, I take that back. The one thing that's not as different, and so these were earlier data. We have more data now from about 20 mice, and we're now seeing that there's actually not much of a difference in invasion between age males and age females. So that's going to be really interesting to sort of tease out because we're seeing so many differences in the growth rates. So just to summarize some of the key changes we've seen, female dermal fibroblasts undergo early replicative senescence. There's elevated ROS in the age male dermal fibroblasts, but female dermal fibroblasts are better equipped with repairing the ROS. And age male fibroblasts promote invasion and therapy resistance in melanoma cells in vitro. However, both age male and age female drive invasion and therapy resistance in vivo. And finally, we're moving beyond melanoma. I know this is a melanoma SPORE talk, so I'll be very quick here, but we're super excited to be working with my dear friend Liz Jaffe. We're co-mentoring Dan Zabransky, who's a very talented hem-onc fellow. And what we've done is to obtain human young and age pancreatic fibroblasts and do a lot of the same assays we're doing and have done with the melanoma cells. And we're also using mouse models of cancer, both having taken young and age pancreatic fibroblasts from these mice and using the transgenic KPC models. And what Dan has seen is he's already started to see some really super interesting stuff. So if he takes pancreatic cancer cells, human pancreatic cancer cells, and treats them with young and age fibroblast conditioned media, he sees that actually they increase their proliferation quite rapidly. They have an increase in invasion. It's not very dramatic in vitro, but when we look in vivo, what we see is in the KPC model that we put into 18-month-old mice compared to 8-week-old mice, the tumors grow out very rapidly in the age mice as compared to the young mice. If he looks at angiogenesis, there's far more angiogenesis in the tumors of the age mice compared to the young mice. And now if he looks at the metastases, he sees that there's far more metastases in general to the different sites in the age mice than there are in the young mice. So that has been super exciting to see these kind of data holding up in a whole different cancer as well. And I'm excited to explore this and tell you more about it later. So I hope at this point, I haven't run over time, and I hope I've convinced you that the aging microenvironment is critical to consider when you're designing your preclinical studies and when you're treating patients. I've told you a little bit about how things like the matrix change, angiogenesis, changes in metabolism, as well as metastasis. And all of these are impacted by fibroblasts, specifically aging fibroblasts. These are the wonderful people who do all the work. I've tried to call them out as I go, but really the lab is a team and works very closely together. I've been so happy here at Hopkins the last couple of years that I've been here, and I have a whole cadre of amazing collaborators, from engineers to immunologists to bioinformatics experts. It's really been a lot of fun. I'm also very lucky because melanoma is such a global effort. We have collaborators all around, like I say, across the street, across the country, and across the world. I usually end with a picture of my favorite aging study, which is my daughter, who is now 16 and a half, almost 17, and looking at colleges, which is just about breaking my heart to think of her leaving. And I will end, as I always do, as Marcus said, promoting diversity and women in science. You know, women, especially women of color, really earn a tiny percentage of even bachelor's degree. And when you think about PhDs, this is an even smaller percentage. If any of you are interested, my dear friend Danita Brady and I have written a commentary in cancer discovery with some actually actionable items and interesting websites and reading material if you're interested in promoting diversity in your local community as well. So I'll stop there and happily take any questions. Thank you so much.
Thanks, Ashley. That was really great. And what I'd like to do is invite folks to post questions in the chat. I don't think we have a Q&A area. So I see one already from Harriet Kluger, and I will read that, and so all can hear. Ashley, thank you for a terrific talk. Do you think we should be looking at old versus young in a binarized fashion and young versus old humans and mice? Do you think there is a difference between old and very old? And that's a real question, Harriet. From my point of view, that changes as I get older, but what does that mean? But anyway, I think I was going to ask a similar question about, you had shown that bevacizumab study, re-looking at clinical data. Should we be looking at all clinical data in trials in a similar manner?
So my bias to that last question, of course, is yes. I do think we should be looking at all clinical data according to age. To answer Harriet's question specifically, although I often present the results as binarized, what we really do is to look at it in bins. So we have our under 50 age group, our 50 to 65, our over 65. To answer your question about old and very old, we call them aged and super aged in the lab. There are definitely differences. And the reason we even started looking at that is because if you look at the incidence of melanoma, right, there's this bell curve where it kind of goes up and up and up. And then suddenly after like 80, 85, the incidence drops off, the mortality rates drop off. And the question is, if you get that old, are you just a super survivor? There's a lot more going on, or is there something actually physical? So for example, one of the physical changes we've seen are that, if you look at the biophysical matrix, for example, there is a bell curve of how a cell can make its way through that. So if you have a super stiff matrix, the cell can only go so far because the nucleus gets stuck. And as you decrease the density of that matrix, the cell starts to be able to move through it. But if you decrease it too far, then it's got nothing to hold on to, and it kind of flounders as if it's in a soup. So that's sort of one of the things that we're looking at in context of age versus super aged. And there are definitely differences. So yes, we do think there's a difference, and we are looking at some of those.
And I'll follow up on that, Ashi. With regard to, there should be a lot of data in this particular area. So AJCC staging for melanoma has been in existence for decades. Mm-hmm. And there are some very well characterized parameters that predict prognosis that are very closely correlated to metastasis. So one of the predictions with some of the discoveries that you've made is that with depending on like the Breslow thickness or thickness of the melanoma, you might have different prognosis in old versus young because they're more likely to metastasize in old. I don't know if you've tried to do something like that already, or what your thoughts are about that.
Yeah, that's a great point, Marcus. So we have not yet tried to do that, only because I think we haven't really had, one of the things we haven't done since we moved here is really fully establish all of the clinical collaborations that we need. And yeah, I would imagine that, I think even with the AJCC staging, age is really the overriding factor, right? So Breslow's thickness is a close second. But it would be really interesting to go back and look at thin lesions in old versus young patients and see if there is an increase in metastasis.
Certainly
right now, things like, Sorry, certainly things like lymph node metastases are dramatically different, where actually younger patients have more lymph node metastases than older patients. But the older patients have more visceral mets. And we think some of these disruptive changes to the matrix, et cetera, are part of that.
It'd be interesting to ask, so like Jeff Gershwell or someone else, who does a lot, who has access to that data, and have them do it, along the way.
It's a great point. I'll reach out to him. That's a great point.
And I guess another question I would have too is that, and you probably guessed this might be a question I might ask, is that it's been kind of surprising that responses for older versus younger patients in melanoma with immune checkpoint inhibitors have been better in older patients. And sort of that also introduces a complication in terms of survival, because there could be immune editing in some older melanoma patients because their immune system might be more primed to fight it. But any thoughts about how the microenvironment might be affecting those enhanced rates of response?
Right. It's a great question. So we think it's several fold. So we published, I think back in 2018, that observation, right, that older patients getting anti-PD-1 do much better than younger patients. And, part of it is that the immune microenvironment is very different between young and age. Actually, younger patients have more Treg, so that CD8 to Treg ratio is off in the younger patients as compared to the older patients. However, our recent data, and data I didn't really talk about today, is how much angiogenesis is playing a role in this. And so, as you know, angiogenesis can have a quote unquote beneficial effect by being a good venue for delivery of immunotherapeutic agents. And so, although VEGF can be a negative prognostic factor for immunotherapeutic delivery, having angiogenesis in the absence of VEGF, which is what we're seeing in the aged, seems to be sort of the sweet spot for the delivery of immunotherapy, which might be a part of it as well.
Super. And it's funny because I think a lot of people had assumed that this concept of immune senescence, in older individuals might be happening, but it might be in specific subsets like Tregs, as you're, mentioning the observation there. So I could go on all day. Oh, here we got a question just when I was getting desperate here. So Brenda Emu has a question. In terms of fatty acid transporters, was FATP2 uniquely upregulated in tumors from older individuals, or is this true for other fatty acid transporters as well, including like CD36, which there has been interest at Yale and as well? And in particular, are there particular lipid species that aged fibroblasts secrete?
So that's a great question. So we looked at CD36, we looked at FATP1 through 6, and the only one that was upregulated according to age was FATP2. Now, that's not to say these others aren't upregulated simply by virtue of these cells being melanoma cells. They are. However, FATP2 is uniquely upregulated with age. In terms of the particular lipid species, yes. So our lipidomics analysis showed us that probably the lipid species that we were most interested in were ceramides. So ceramides seem to be the most differentially expressed or secreted by the aged fibroblast, the most efficiently taken up by the melanoma cells in that environment, and the ones that have the most impact on, that wasn't in the cancer discovery paper, but we've shown that ceramides can drive invasion and metastasis in melanoma cells as well.
Interesting question. Interesting. It's really interesting results. Last chance for folks to ask questions. This has really been a wonderful summary, especially for us at Yale, as we're interested in getting an aging center set up. I had emailed a mutual friend of Ashi's and mine, so Deep. I don't know if he was able to make it.
He's in India right now.
Oh, is he? Okay. I won't kick him in the shins too hard then. But anyway, obviously a really great interest for us here. Jeff Townsend also has a question. I think I saw 3 fibroblast lines used for at least one comparison, and in that comparison, results were very consistent. are there any inconsistencies among different samples or lines?
Sure. So Jeff, of course, we, for most of the experiments we've done at this point, we've used up to 12 or even 15 in one case, different cell lines. There are definitely some inconsistencies. The most consistent inconsistency that we see is that we have some young lines that behave as if they're an old line. And when we go back and sort of dig through the history of those lines, they tend to be from young women with a tanning history. So that's another ongoing project in the lab I didn't talk about, is you keep looking at the effect of UV as a premature aging agent. So wear your sunblock, everybody, but you already know that.
One question related to that is that there's, including some work from Yale, interest in looking at DNA methylation and epigenetic changes as an aging clock. Yep. And have you done any looking into as to, in those cases where there's a disconnect between chronological age and biological age, if there's a component related to DNA methylation? That might be UV induced, right?
Right. So in terms of melanoma, we're just beginning to delve into that. However, we are collaborating very closely with Hari Iswaran, who is an associate professor here at Hopkins, who works on colon cancer. And we've got a lot of interesting data coming out of those studies. So I'm super excited about that. And of course, people like Jean-Pierre Issa and Shelley Berger have done a ton of work in, understanding this epigenetic drift that we see during aging.
Well, super. Well, unless we have another last minute question, I would really like to thank you, Ashi, for giving us such a stimulating talk. Obviously generated a lot of interest. We're looking forward to actually having you here in person sometime in the future. But thanks so much for sharing all of your work. And I would encourage folks to also read that Cancer Discovery article about increasing opportunities for scientists of color and of other backgrounds that are less advantageous to move forward. So thanks so much, Ashi.
Marcus, thank you so much for having me.
Absolutely.
And hope
Take care.
See you soon. Take care.